I would say that it's an osmoconformer. Okay. Other organisms, right, like this fish, right, this organism does not conform. It needs to maintain its concentration of water and salts at a different concentration to that of the ocean, and therefore this organism would be a regulator, and since we're talking about the regulation of salts, I would call it an osmoregulator. Okay? All right. Osmoconformers and osmoregulators. Now, we can talk about the range over which these organisms are doing this conforming or regulating, right? That sea star, even though it's a conformer, it cannot conform over very broad ranges, right? I could take 3 gallons of, or 30 pounds of salt and dump it on this sea star. I'm going to kill it because it cannot conform above a certain concentration of salts. If I put it into fresh water, it's going to die because it can't conform below a certain concentration of salts. Same thing with my fish. There should be some limit on the high end of salt concentration, on low end of salt concentrations, where its regulatory systems cannot keep up with the movement of water as a consequence of differences in concentration between the fish and the environment. At some point, the regulatory mechanisms don't work anymore. They can't keep up with the problem. So we're going to come up with some other language here to describe the range of salt concentrations that we can deal with. So narrow ranges, the prefix is steno, which means narrow. And then haline, because you can never have enough words that mean salt. So stenohaline is an organism who's got a very narrow concentration of salts that it can exist in. I could be an osmoconformer who's stenohaline. I can also be an osmoregulator that's stenohaline. And then organisms that can exist over broad ranges, these are referred to as euryhaline. This is wide. And then, of course, haline is salt. So euryhaline organisms exist over a broad range of salt concentrations. So I could have a sea star species that exists in the ocean. In the particular ocean it lives in, it doesn't get exposed to tides all that much where it lives. It's not close to the outflow from a river. And that species may be stenohaline, right? It conforms, but it conforms over a very narrow range. And through long-term adaptation, its environment doesn't range that much, and therefore its concentrations that it deals with aren't all that broad. So it's a stenohaline conformer. Same thing with a fish that lives in that particular location. That might be an osmoregulator who's stenohaline, right? It's not used to dealing with these fluctuations historically as a species. It doesn't have these mechanisms to deal with these large fluctuations. On the other hand, there are sea star and fish species that live in estuaries. An estuary is a place where fresh water and salt water meet and often mix. So you get the tide coming into the estuary, salt concentrations go up, the tide goes out, saltwater concentrations go down. And it's more dominated by the fresh water. So for these kinds of organisms that live in a place like an estuary, eurohaline is what they need to be, because over the course of 6 hours I'm going from high to low tide. I'm experiencing these rapid changes in salt concentration in my environment. Okay? All right, so I've got, as I said, I have some of these out of sequence, so let's skip that other slide. We'll come back to it. Okay, all right, so let's start with a few examples. Okay? So, again, I know most of you are human-focused. Humans are complicated. Our main osmoregulatory trick is this thing called the kidney, which is brutally complex. Right? You can specialize in the kidney. That's how we know it's pretty complex. But as I've said before, I think it's easier sometimes to look at more simple mechanisms out there in the animal world, get a handle on how things are handled at a simple level, deal with some of those issues, and then layer our way up, build it at becoming more and more complex until we get to the most complex thing, the kidney. But we've dealt with all these kind of minor issues, and I think sometimes it's easier to put it all together if we deal with some of these more simple organisms. So we're going to start out with the bony fish here first, and let's start out here with a fish that lives in make this a little bit better looking fish than I did before. All right, so here's my fish. Let's put it into seawater. Okay, so we're dealing with a fish that's a saltwater fish. That's the one on the right there. Okay? Now, the problem for this fish living in seawater is that the fish itself is actually a lower salt concentration than its environment. So its osmolarity, I would refer to the fish here, is hypoosmotic, and its environment is hyperosmotic. The concentration of salts out here in the seawater is much higher than it is in these bony fish. It's just the way they are. Now, because of this difference in salt concentration, and because salts don't freely move from organisms into the environment because of this whole membrane problem, right? You have to leave a cell by crossing a membrane. Salts don't do that very well unless we have protein channels or carriers, but this automatically sets up a problem in that salts don't move, but water does. Which means if the environment is hyperosmotic and the fish is hypoosmotic, then water is going to want to leave the fish through this process called osmosis. Water is leaving the fish because of differences in salt concentration. Okay? So that sets up the problem, right? The problem is the fish is losing water to its environment, leading to a very real problem here of dehydration. Right? It seems kind of counterintuitive, but this fish can dehydrate in seawater. Okay? It's going to dry out, losing water to its environment. So we got to think of some ways that this fish is going to be able to deal with this problem. Okay? Now, of the water that's leaving the fish can protect itself from water loss in a few different ways. One is, right, I can coat myself in mucus that will prevent water from moving across me very easily, and fish do that. If you've ever held fish, they're very slimy. Slime helps with other things. It's an immunological barrier, but it also functions to prevent water movement across the integument. But the problem for the fish is it does have a structure here, right, these gills, that you can't protect them from the movement of water because we're trying to encourage the movement of oxygen and CO2. Along with that, water is going to move freely across gills. They don't have a lot of structural tissue. So if you see up here, it says water leaves gill capillaries and enters seawater through osmosis. Just the way it is. Right? Most of the water lost by this fish is going to be lost across the gills. Okay? Now, as you lose water, you have kind of two problems running at the same time. The first is dehydration of the cell, and the second is increased concentration of salts. Because if I take any fluid and it's got some salts in it, and I remove water, I've concentrated the salts. Okay? Now, dehydration is an issue for cells. I think we've talked about this before, but just in case we haven't, water is a problem for cells, in particular for protein shapes. We know that proteins have to be in particular shapes in order for these proteins to function properly. The shapes that they are in, the shapes are encouraged, right? These shapes are encouraged by interactions of R groups with each other, as well as hydrogen bonding between the chains, etc. But some of these R groups are interacting directly with water, and some of the R groups in here are interacting with each other to avoid water. So the water is helping to maintain the three-dimensional shape of this protein. It's keeping it locked into a particular shape so that it can function in a particular fashion. As I remove water, I concentrate the salts. Right, this is a slightly positive charge. Maybe that's a negatively charged amino acid. This interaction is encouraged in an aquatic environment, the aquatic environment of the cell. But as the water is leaving, the salts that are present can also interact with this protein. And if the proteins R groups are interacting more often with these ions than they are with water, then what happens is the protein is no longer dissolved in the fluid of the cell, and it goes through a process that we call salting out, meaning the protein jumps out of solution. Right? Those of you that have ever played around with starch, right? You can add cornstarch to kind of thicken up stuff, maybe make some gravy from those turkey juices in the pan. That act of trying to get that starch molecule into the water is tricky. If you just dump the starch into the water, it won't dissolve in it. Why won't it dissolve? Because the starch molecule has lots of charges all over it too, and it's interacting with itself rather than with the water. It doesn't want to dissolve in the water. So a similar process happens with these proteins. If they interact with ions more often than water, they salt out, they come out of solution, and just like trying to get cornstarch into water, you're not going to be able to get these proteins back into solution. And if they're not in solution, they don't interact with substrate properly, they don't interact with other molecules properly, and that's junk protein now. So the two issues here are losing water from the cell and a concentration of ions within the cell. So this fish has to deal with both problems. Okay? So one of the problems is the loss of water. Well, how do you get the water back? Well, organisms don't move water. The trick is instead adjust the osmolarity or the tonicity, and the water will move appropriately. So if I'm trying to get water to move into the fish, one solution would be bring in lots of salt, and then the water will start to diffuse in. That is, make yourself hyperosmotic to seawater. Then the seawater would have a lower salt concentration, and water would always be coming into this fish. Unfortunately for the fish, that's not a solution, because the salt concentration in cells would become too high, and then we would have those problems of salting out, etc., etc. So the other way the fish is going to deal with it is to drink seawater. Just drink it. So you open up your mouth and you drink as much seawater as you possibly can. Now, you're also going to get water from whatever food you eat. Okay, so if I go around eating other fish that are hypoosmotic, I'm getting water at the proper concentration. On the other hand, if I eat something like a sea star, who's isoosmotic to seawater, I'm increasing the problem, because I'm taking in an excess of salt relative to the amount of water that I need. I'm doing that in the food that I eat. Okay? So the way you do that is you drink seawater. Well, when you drink seawater, you're also taking in the ions that are found in seawater. So the solution of losing water is to drink it. But now that's introduced another problem. How am I going to deal with all these excess ions that I've brought in? So, a couple of solutions. The first is, let's do a lot of active transport, and let's actively transport salts out of the fish into the environment. So I'm going to actively transport salts out of the fish into the environment, and I'm going to do that in a location where it's easy to transport, and that's the gills. So here, across the gill epithelia, active excretion of sodium and chloride. So the fish is going to be drinking water and then actively transporting out the salts into the environment. That's osmoregulation. I'm adjusting the concentration of salts and water in me. The next thing I can do is use my kidney to play the same game. I'm going to actively transport salts into my urine, and then here, small amount of concentrated urine containing large amounts of salt. So I've got this kidney trick. I'm going to push salts into my urine and then excrete them out. Okay? So osmoregulation and excretion are kind of interconnected. Right? This is just osmoregulation at the gills, but what's happening inside the fish at the level of those kidneys is I'm taking and doing osmoregulation and coupling it with the excretion of these salts. Okay, so the systems, osmoregulatory and excretory, are kind of running together at the same time. Okay, so if I'm lower concentration of water or salts than my environment, right? If I'm lower concentration of salts, yes. That means higher concentration of water. Water leaves me. I drink the water to get it back, but I bring in the ions at the same time. I need to actively transport the ions out of the body. I do